Hi, my name is Caitlin Bowen. I was a graduate student with Dr. Hal Dietz at the Johns Hopkins University School of Medicine. Today, I'll be presenting a gene variant and mechanism for a potent protective modifier of vascular Ehlers-Danlos syndrome. So we have two genetic models of vascular Ehlers-Danlos syndrome, or VEDS, one with a glycine substitution G209S and one with the substitution G938D. Both our mouse models recapitulate VEDS phenotypes. So these mice die suddenly due to aortic rupture, aortic dissection, or organ rupture, most often presenting with a rupture in the thoracic aorta, which is shown in the center. So both of these curves show survival curves, where the percent survival is on the y-axis and the age in days is on the x-axis. So the faster a curve falls, the worse the survival. So mice with the G938D mutation, shown in orange, have a more severe phenotype. They have a median survival of only 45 days, and 100% of them die by 6 months of age, whereas we see a median survival of about a year for our G209S model, which is shown in blue. So going forward, I also refer to these as the mild and severe models. So previous work has gone on to show that this signaling pathway called PLC-PKC or ERK pathway is activated in vascular segments of these mouse models. And if we attenuate this signaling axis or inhibit it using molecules like hydralazine, enzastaurin, or cobimetinib, this affords overt lifelong protection in these mice. But in this study in particular, we sought to understand the variability in phenotypes, or why some patients with VEDS have events early in life and others can go for decades before having an event. This variability in genetic diseases can be explained in part by allelic heterogeneity, such as the fact that patients who have null mutations have less severe phenotypes than those who have missense mutations. But you can also see variability in people that have the same mutation, or even people within the same family. And these differences could be explained by differences in environmental factors, or also genetic variation in other genes, which we will call modifier genes, because they can modify the phenotype. So mice are actually really good at being able to use to identify these modifier genes because mice are genetically identical. So different strains or backgrounds of mice can be utilized because we can control for both the allele and environmental factors. So we use two strains called 129 and Black 6. These two strains of mice are nearly genetically identical to each other, but have a few genetic differences that can lead to different phenotypes. So for example, some of these differences lead to differences in coat color, where the 129 mice are brown and the Black 6 mice are black. So with this idea of genetic modifiers in mind, we introduced our VEDS mutations onto pure Black 6 and 129 backgrounds and looked at the difference in phenotype. So for both our mild and severe mutations, they're associated with early death due to aortic rupture on the Black 6 background, as shown in black. These are the same curves we looked at before. But remarkably, the same mutation shown on a 129 background, which is shown in red, leads to essentially complete protection from vascular catastrophe and premature death, where these mice are nearly 100% of them are alive, even after a year or 6 months of age. Informatively, these differences in survival were not attributable to differences in blood pressure between the two mice or differences in the actual mechanical strength of the aorta. These 129 mice did not have more intact collagen, they didn't have more intact elastin, and they actually looked exactly the same as the susceptible VEDS mice, or those that are on a Black 6 background. So this unanticipated dissociation between the actual tissue integrity and the risk of rupture was really quite striking. And it led us to hypothesize that potentially there was something in the cells rather than the matrix that determines vascular disease risk in VEDS. And if that were true, it would really bode well for the identification of interventions that target this cellular response. And informatively, mice that are 50% Black 6 and 50% 129, which is shown in blue, have improved long-term survival. So we move forward with the hypothesis that the 129 background has a variant in a gene or genes that can rescue the risk of vascular rupture in VEDS. So to identify this modifier gene, we used survival time to stratify mice into cases and controls, or mice that die from aortic rupture before 8 weeks of age, which is similar to our severe background, and mice that are long-term survivors. So when we mix these mice together, we get a mixed background cohort of EDS mice that have a random assortment of BLAC6 and 129 alleles. And informatively, this mixed background population, which is shown on the right, does stratify into these two case control or susceptible and protected phenotype groups. So we asked, what genes does this group have that this group does not? And we did that using a genome-wide association study. So this Manhattan plot shows the negative log of the p-value on the y-axis and all the different locations in the mouse genome on the x-axis. And this red line indicates genome-wide significance. And so you can see there's one dot that associates with protection. And the odds ratio at this locus is significantly less than 1, which means that if you have a 129 version of the allele at this locus, you're protected from death due to aortic rupture. So we then asked, what is the gene that's at this locus? So in the region that's surrounding this SNP from the previous graph, there are 102 genes. Only 31 are expressed in the aortic wall, and 4 of those were different between BLAC6 and 129 backgrounds, but only 3 had predicted functional differences between the two backgrounds. And really one stood out as being particularly relevant, and that gene is called MAP2K6. So MAP2K6 has a variant that exists between BLAC6 and 129 that's predicted to affect the function of the gene. So then to ask if this gene was really relevant to VEDS, we've knocked it out in the protected 129 VEDS mice or took it away. And we found that if you take away MAP2K6, they are all of a sudden susceptible to death from aortic rupture. And this indicates that the function of this gene is really critical for the protection that these 129 mice have, confirming that this could be a major modifier in our mouse models of EDS. So we then asked, how might MAP2K6 affect the risk of aortic rupture in VEDS? This gene encodes a protein kinase called M2K6, which activates another kinase called P38. P38 then acts directly on protein phosphatases PP1 and 2A, which are main phosphatases that dephosphorylate or deactivate both PKC and ERK. So we therefore hypothesize that this protected 129 background has increased MAP2K6 activity, leading to increased P38 activation, increased protein phosphatase activity, and therefore a reduction in how much PKC and ERK are activated, which would protect them from disease. And just to go quickly, as we expected, we saw increased P38 activation, we saw increased protein phosphatase activity, and we saw decreased PKC and ERK activation. To then show that P38 was really the important gene here that's downstream of MAP2K6, we treated mice with a P38 inhibitor and found that this increased the risk of vascular rupture, but that if you protected them by also treating them with a MEK inhibitor, which inhibits ERK, you can rescue the risk of vascular rupture. And then to show relevance of MAP2K6 to this PKC-ERK pathway, we treated those 129 VEDS mice that did not have MAP2K6 and so were susceptible to disease. We treated them with a PKC inhibitor and showed 100% rescue after 3 months of treatment, compared to only 60% survival in untreated mice. So really taken together, these data illustrate that the 129 background is able to prevent vascular rupture because it has a variant in MAP2K6 that increases P38 activation and protein phosphatase activity, leading to normalization of PKC and ERK. And if you inhibit this protective axis in EDS mice, you increase the risk of rupture, but this occurs in a PKC and ERK-dependent manner. So quite remarkably, this rescue on the 129 background isn't dependent on an improvement in collagen deposition or any changes to the inherent structural integrity of the aortic wall, but instead appears to be dependent on the biochemical and response of vascular cells. So this bodes well for the development of pharmacologic strategies that can mimic this mechanism of protection. And it also adds confidence to the potential of therapies that target this PKC-ERK axis. These findings justify a clinical trial of a PKC inhibitor like enzastaurin, which is a study that will begin recruiting at the end of the year. So these results both validate and extend our understanding of cellular signaling events in VEDS. And with that, I would like to thank all of those who worked on this project, as well as our funders, and you can always email me with any questions.